sir.
Yes, sorry.
C-section.
C-section. Okay, you're foreigner.
Yes, sir.
Okay, wonderful. You're from Nepal?
Ji, sir. Nepal.
Okay. How many students admitted in this semester or in your time?
5 in
Five,
this,
five Nepalese students. Yeah.
sir.
Okay. So you are attending the class from the Nepal. Is it right?
Yes, sir.
Okay, wonderful. So today we will go through a chapter. Did you heard about euchromatin, heterochromatin?
No, sir.
Not taken in this class
No, sir.
in this section.
No, sir.
Okay, okay,
এগুলো
no
সে
problem.
বলছে এমনি পড়ছে
Sorry.
একটু হালকা হালকা বলছে স্যার এমনি আর কি
ও হালকা হালকা করছে
এখানে
আচ্ছা এটা ক্লাসে নেওয়া হয়েছে তোমাদের
না
ইউক্রোমাটিন
স্যার
হেটেরোক্রোমাটিন নেওয়া
না
হয়নি
স্যার
okay no problem I will carry on this class I already discussed this lecture in A and B section today I will go through on these topics to you
স্যার আপনি কি শীট প্রোভাইড করবেন স্যার শীট দিবেন কি
yes at the end of this class I will deliver this sheet to Zerin Sama maybe and Rupam. I will at the end of this class I will email to this student. Okay, you can
স্যার
see my screen maybe. Can you see my screen?
ইয়েস স্যার
আমার স্ক্রিন দেখা যাচ্ছে?
ইয়েস স্যার
Okay, wonderful. So okay, I need to decrease it. Okay, in the screen we can see there is euchromatin, heterochromatin, heteropegalesis. Okay, actually in this name you can see chromatin and heteropignosis. Yes, so chromatin, we need to know about the chromatin. So you know that chromosome, right? Okay, just a moment. হ্যাঁ রতন হ্যাঁ কোথায় হ্যাঁ রতন ভালো রতন তুমি কই okay you know that chromatin yeah say normally chromatin in the interphase stage when the chromosomes are thread like structure actually it's found on the interphase and we call this chromatin or chromatin fibers the composition is it's composed of nucleic acid and protein that's why the nucleic acid part is the DNA, nucleic acid part is the RNA, deoxyribonucleic acid, ribonucleic acid, and protein, histone protein, non-histone protein. and which condenses to form chromosome during cell division. During cell division, it's condensed, and we can tell that these are chromosomes. So where it can be found? So it can be found in the nucleus, and in case of eukaryote, and in case of prokaryote, it can be found in the nucleus. So in prokaryote, we can found the nuclear membrane. That's why the nucleus or nuclear material or DNA can be exposed in the cell. So this known as nuclear, and it takes colors. So that's why it can easily recognize through staining. Therefore, its name literally means colored material. Chromatin, chromosome, colored bodies. Chroma means colored, some means bodied. That means chromosome means colored bodies. So chromosome derived from chromatin. So that's why it takes colored. That's why its alternative name is colored material. Okay. So it has two types, heterochromatin and euchromatin. And these two names are given by two scientists. One scientist, his name is Hayes in 1928 to 29. He has given this name heterochromatin and euchromatin. Although this has been described earlier, but Hayes has given the euchromatin and heterochromatin in 1928. And in the picture, if we see, if it's in the cell, we can find the mitochondria in the in the side, and in the cell, you can find the nucleolus, you can find the chromosomal materials, you can find the nuclear membrane, and in the inside of nucleus, you can find mainly two types of material. One is darkly stained, which remains at the peripheral regions, and another one is lightly stained, which remains dispersed around the across the nucleus. So the lightly stained or the white substance are known as euchromatin, and the dark stained materials, the black one, is known as heterochromatin. Okay. Now I will go through the two types. One is heterochromatin. Heterochromatin is the area of chromosome which are intensely stained. Intensely stained means deeply stained. Stained means taking the color with DNA specific strains and are relatively condensed is known as heterochromatin. They are tightly packed form of DNA in the nucleus. So in the nucleus, scientists have found that some chromosome takes deep stained. Some chromosomal regions are deeply stained, and they are tightly oriented in the nucleus. So these are known as heterochromatin. So there is a problem occurs due to high compactness, or the problem creates, and the uh, relationship with the function due to the high compactness. If you go through a picture, you can find that this coiling portion of the nucleic acid, the blue one. Yeah, in the left hand side, you can find the euchromatin. In the right hand side, you can find the chromatin. These are taken from the chromosome. There is a bichromated nature chromosome. And if you take a portion of this, then you can find the structure like this. So you can see the proteins 1, 2, 3, 4, 5 histone proteins are compactly arranged. On the other hand, the three in the same, if the length is same, in the same length, the three histone proteins are found and the DNA are loosely composed. Okay. So due to its high compactness, there is a problem creates in the function. The organization of heterochromatin is highly compact in the way that these are inaccessible to the protein which is engaged in gene expression. Okay, due to its high compactness, actually protein does not form. Actually, we know that there is a central dogma. There is a central dogma in the molecular biology. What is it? It's DNA, then mRNA, and then protein. DNA, mRNA, and protein. Firstly, transcription and then translation, and then protein is formed. Actually, this cannot be happen due to the high compactness or tightly packed heterochromatin. Even the chromosomal crossing over is not possible due to the above reason. So due to its high compactness, they not lose, they do not share the portion of the chromosome with the male or female counterpart. As a result, crossing over is not possible, resulting them to be transcriptionally as well as genetically inactive. Transcriptionally, that means the protein related function, and the genetically active, that means the crossing over function. Yeah. Okay. This is the problem. This might come as a short question. What the problem creates in the function due to the heterochromatin? Okay. Let us study the types of heterochromatin. There is two types of heterochromatin. One is facultative, and another one is constitutive. And facultative, actually, it's create some. This creates a problem in the function. The first one is the RNA production. These genes which get silenced through the process of histone methylation and small interphase RNA through RNAi are called as facultative heterochromatin. Hence, contain inactive genes and not a permanent character of every nucleus of the cells. Actually, these get silenced. They do not take part in uh, RNA synthesis or histone methylation. So that's why these these remain inactive, and these genes are called inactive genes. And the second one is the constitutive. As constitutive is the DNA which is responsible for the extracellular function of genes. Here we can find that while the repetitive and structural function genes like telomere or centromere are called as constitutive heterochromatin, and this can be found in the telomere or centromere. That's already you have studied in the chromosome morphology. Maybe Bhavin sir or any madam already taught in that class. Okay, this structure is retainable during the interphase of the cells. So you can find this in the interphase. Okay, overall function of heterochromatin, to protect the DNA from the endonuclease damage, it's due to its compact nature. Actually, endonuclease is an enzyme which break down the chromosome, which cleave the DNA. So this is protected through its high compact nature. And another function is the prevent the DNA regions to get access to proteins during gene expression. Okay, it also prevents the protein formation. We already discussed this in the previous slides. Now I'm going to to the second type of chromatin that is known as euchromatin that's found lightly stained material in the nucleus. The part of the chromosome which are rich in gene concentrations and are loosely packed form of chromatin is called as heterochromatin. They are active during transcription. So in the previous one, the heterochromatin one is not active in transcription or they are not friendly in transcriptional activity. But euchromatin are active in transcription. That means it can produce from DNA to messenger RNA, from messenger RNA to protein, or you can say that amino acid. Okay. Euchromatin covered the maximum part of the dynamic genome to the inner of the nucleus, and it is said that euchromatin contains about 90% of the entire human genome. So if we go through the picture, in the picture we can see that whitish part, the white part, it contains the maximum portion of the nucleus. So that's why it says that it can cover around 90% of the genome or human genome. Okay, now I'm going to the function. Euchromatin actively participate in the transcription from DNA to RNA, and it encodes the functional protein. So, and you can tell that it's also activates, it's also takes parts in the crossing over. You can add some more functions. Okay. The structure, if we see, the first one is the euchromatin. You can see the structure, there is 3 and there is 5, yes. And the blue one is the DNA, the circular one is the histone, and acetyl group you can find here, A, the purple color, with the P is a phosphoryl group, and M is the methyl group. Okay. Okay, this is another picture. You can draw any picture like this one or the previous one that I showed earlier. So I tell you again that the dark stained material is the heterochromatin and the light stained are the euchromatin. Now the difference, we already discussed this issue, but it should be when you will write a difference, it might have the basis of comparison, and through the basis you should write, you should differentiate what's the character present here, what's the character present in another one. Okay, the first is the overall meaning. Overall meaning is that the tightly packed form of DNA in the chromosome is called as heterochromatin, and the euchromatin are the loosely packed form of DNA. DNA density, obviously, it will be higher in the heterochromatin. We found at the dark region. So there is a high density, and there is low DNA density. And in the context of staining, they take stain dark and they are takes light stain. That's why I write here lightly stained. And where they are present? They are found at the periphery of the nucleus in eukaryotic cells only. They are found in the inner body of the nucleus of the prokaryote as well as in the eukaryotic cells. Okay. So it's clear maybe where they are present. They are found at the peripheral. That means around the nuclear membrane you can find here. So these are found in the inner body of the nucleus. Transcriptional activity. They show little or no transcriptional activity. They actively participate in the process of transcription. Yeah, somebody, Lipun Kumar, raised his hand. So I will discuss this issue just after finishing this one. I will give some time to discuss this issue. So then we'll discuss, yeah. transcriptional activity show little or no transcriptional activity. They actively participate in the process of transcription. Actually, it's true that actually they are not friendly in transcriptional activity or to formation of to form protein, but they are friendly to formation of or to initiate the transcription or translation process. Okay, other features, they are compactly coiled. About the coiling, they are compactly coiled, they are loosely coiled. As we have seen that 3 and 5 histone protein and the coil also. They are late replicate, they are early replicate as they are in a favorable condition, eukaryotic, and they starts the uh, replication earlier, and the heterochromatin are tightly packed. That's why they take some time to reorganize, and then that's why they are rate replicative. Regions of heterochromatin are sticky. Regions of euchromatin are non-sticky. These are the properties. So that's why I'm not going more about this. They are genetically inactive. They are genetically active. Genetically inactive means they do not take part in the crossing over. They do not take part in protein synthesis. So that's why they are genetically inactive. They are genetically active. Phenotypes remain unchanged of an organism. For this, let's say father, then son, then grandson comes. So due to this nature, heterochromatin, the phenotype remain unchanged from one generation to another generation. And in heterochromatin, variation may be seen due to the effect in the DNA during the genotypic process, during the genetic process. Actually, it can create crossing over. It's participate in transcription. So that's why variation may be seen. Okay, these are the difference. So this is an important question about the difference between heterochromatin and euchromatin. Okay, heteropignosis. Heteropignosis is another term. Actually, it is related with heterochromatin and euchromatin. Actually, heteropignosis. During heterochromatin formation, you have seen that this takes deeply stained. So this deeply stained nature is denoted as heteropignosis. It is a phenomenon shown by chromosome in which the certain regions of chromosomes are stained more intensely than others. The regions which exhibit heteropignosis may be said to be composed of heterochromatin as described by Hayes. So Hayes, according to Hayes, uh, the heterochromatin regions, we know that it takes more dense, and it takes more stain than the euchromatin. So that's why the heterochromatic region are denotes also as heteropignosis due to its color taking activity, and called heterochromatic parts against the other parts. So heterochromatin is one side, and in another side the other parts which never show heteropignosis or do not take much more stain are known as euchromatic parts. There is two types: positive heteropignosis, negative heteropignosis. Actually, positive heteropignosis, actually the heterochromatic regions which may be thicker and takes more stain than the euchromatic ones. These are known as positive positive heteropignosis. And the negative heteropignosis, in this case, the heterochromatic regions may appear thinner and stain more weakly than euchromatic regions in the same nucleus. Okay, these are the weak stain, and these are the thinner part. So that's why these are known as negative heteropignosis. Okay, so now this portion has been, this is a small topic, but one question will come from here. The student, we can discuss it. So if you have any question,
স্যার
then discuss. Yes.
স্যার প্রথম থেকে স্যার একটু মিস করছে স্যার আমি স্ক্রিন রেকর্ড করছি স্যার আপনি যদি একটু আরেকবার করে স্লাইডগুলো দেখাতেন স্যার আমার স্লাইডগুলো স্যার আবার খুলে আমি
Actually, there is, I will tell you that there is no need to show these slides because I already give you the PDF format. So don't need to be worried about this. Yeah. But if you want, I can show you just for recording. This is the first slide, the topics of this lecture today, and then chromatin, and then I discuss about what is the chromatin, where you can find, and how this name come as chromatin or chromosome. And then the picture, overall picture, in the nucleus picture, you can find that euchromatin and heterochromatin, and then the types, and then who has given this name, and then heterochromatin. I described about heterochromatin, and then there is a problem creates in the transcription and function due to its nature. I already explained why this happens, why this are genetically inactive or transcriptionally inactive. And then I go through the types of heterochromatin. There is two types, facultative and constitutive. Actually, facultative, these are the genes which get silenced. And constitutives, actually, they takes part in the structure, like these are found in the telomere and centromere. And then I go through the function of this, and then I go on another major type, euchromatin. I said it about it and how much percentage it covers in the genome. Then I told about the function of this. I showed a picture how the structural part is organized. So we can find here euchromatin as loosely arranged. On the other hand, the heterochromatic parts are tightly arranged. You can see the compactness of this through the coiling and the structure of histone protein. And then I repeat another figure of this. It's a nucleus picture. You can find the darkly stained heterochromatic region and lightly stained euchromatic region. And then I go through the difference between euchromatic and heterochromatic region. Initially, uh, some bases with regard to some bases, and then containing the other features. And then the last part was heteropegalesis. What is this? Actually, normally this comes through the staining properties of heterochromatin. Normally, the heterochromatin regions staining properties is known as heteropegalesis. So then I went the types of heteropegalesis. So this is all about the lecture today.
থ্যাংক ইউ
Okay, no problem. And now I will send this. I will send this to my students. So, Jerin Saima, are you present here today? Jerin Saima.
Sir.
Okay. So I will send one copy and Rupam, the seer from voice part. Yeah. Okay. Jerin Saima. Jerin Saima60@gmail.com, and the Rupam. Can you tell your ID, please?
118 স্যার।
Huh?
Email.
Email ID.
রেজনুল মল্লিক রূপম।
I got you.
রেজওয়ানুল মল্লিক রূপম।
Is it right? Okay. Did you get the email yesterday?
Yes, sir.
Okay, no problem. Just I'm sending it. So this is a small chapter, and I have sent it to you through the email. The other student should share this PDF from him. Okay, and I have seen the syllabus yesterday. It's a small syllabus. We have seen that most of the parts already taught in the class. So Bhavin sir is taking the major portion of this, and Professor Hasanujaman, he will come soon. He will take physical and chemical agents of chromosome. Do you know the syllabus of this? Level 2 semester 2. It's a very small syllabus. Can you see the screen? Okay, maybe. Okay, can you see in the screen?
Yes, sir.
Can you follow the screen? So this is your syllabus, yeah?
Yes, sir.
This is your syllabus. So cell and cell constituents is already studied by Shohana Joy, lecturer. And then cell cycle and important events of mitosis and meiosis. Maybe Shahnaz Turin plus Bhavendra Kumar Bishwas has already discussed this, yeah?
Sure.
And then chromosome morphological and chemical composition. So this already discussed by Shohana Joy, maybe?
Yes, sir.
Okay, then heterochromatin, euchromatin, this already discussed today. Then special types of chromosome. As I remember, Professor B.K. Bishwas started this one last week. Special types of chromosome: polytin, lambda, sex, beta chromosome, isocentric chromosome, diploid chromosome, etc.
Yes,
Is it
sir.
right? He started this. Okay, he already studied the karyotype, as I
Yes,
remember.
Yes,
Is
sir.
it right?
Yes, sir.
Okay, so these types, effects of different types of physical and chemical agents of chromosome, this is already not discussed. It will be taken by Professor Hasanujaman Rots. Okay, in your practical parts, if you tell, practical parts can be possible when the lab will be open. So without this, we can't practice in the online. But we can discuss, if you're more interested, then we can discuss only the theoretical portion of this, and the job tasks, obviously or must, you have to attend the laboratory. Otherwise, you can't, the things will not be clear. Okay, students, any questions from you? So around 90 student presence here in this class. So did you understand what I delivered in the class?
Question.
Yes, please. হ্যাঁ বলো, বাংলায় বলতে পারো, সমস্যা নেই। কই কাউকে কেউ আনমিউট করে দিয়েছে নাকি? প্লিজ বলো। আচ্ছা, আমার মনে হচ্ছে
নেটওয়ার্ক চলে গেছে।
হেটারোক্রোমাটিনটা স্যার
হেটারোক্রোমাটিনটা ইনঅ্যাক্টিভ থাকে। হ্যাঁ শুনছি, হেটারোক্রোমাটিনটা
এটা কারণটা একটু বলতেন স্যার।
কারণটা হচ্ছে যে এখানে হাইলি কম্প্যাক্ট থাকে, যে ইয়াগুলো থাকে, কি বলে হচ্ছে যে এখানে তোমার হচ্ছে যে এখানে যে তোমরা কম্পোজিশন যদি পড়ে থাকো ইউক্রোমাটিন হেটারোক্রোমাটিনের, তাহলে সরি ক্রোমোজোমের যদি কম্পোজিশন পড়ে থাকো, সেখানে হচ্ছে যে এটা হচ্ছে যে দুই ধরনের প্রোটিন থাকে, একটা হচ্ছে হিস্টোন প্রোটিন, নন হিস্টোন প্রোটিন। তো হিস্টোন প্রোটিন আর নন হিস্টোন প্রোটিন গুলো এর সমন্বয় এবং ডিএনএ খুব টাইটলি অ্যারেঞ্জ থাকে, যার ফলে এটা হচ্ছে যে কয়েল থাকে না, এবং কয়েল যদি না থাকে তাহলে ডিএনএ গুলো মুভ হবে না, ঠিক আছে? ডিএনএ গুলো যদি লুজ না হয়, তাহলে হচ্ছে যে ফার্দার ডিএনএ থেকে যে আরএনএ ফরমেশন, প্রথমে ডাবল স্ট্যান্ডার্ড থেকে সিঙ্গেল স্ট্যান্ডার্ড আরএনএ হয়, ঠিক আছে? তো তারপরে সিঙ্গেল স্ট্যান্ডার্ড আরএনএ এটা প্রোটিন ফরমেশন করে, অ্যামিনো অ্যাসিড প্রোডিউস করে। তো আগে কয়েল গুলোকে তো আগে লুজ হতে হবে, আনকয়েল হতে হবে। তো এটা যদি টাইট ভাবে যদি কোনো কিছুকে প্যাকেট যদি রেখে দাও, ওটাকে যদি তুমি গলাইতে না পারো, তাহলে ওটা তোমার ওই প্যাকেটটা ওরকমই থেকে যাবে, শক্তই থেকে যাবে। এই জন্য আগে তোমাকে ইয়াগুলো সরিয়ে দিতে হবে আগে, ওকে? আনকয়েল হতে হবে, ঠিক আছে? লুজ হতে হবে, এটাই। or any question?
Yes, thank you.
Okay, so hopefully you have a nice attention in my class, because I did not get muted to all. All students know about the techniques, how you will attend a class, and when a noise comes from outside, or it creates a problem to the other students. So that's why it is necessary to get silence. Yeah. Okay. So thank you so much. So hopefully we will see in the next time. We don't know, we are not sure when we will physically see each other, because due to COVID-19 situation, at present its condition is a bit better compared to the last month. So the intensity of COVID-19 percentage is getting lower and lower day by day. So hopefully this will be continue, and we will pray to our God that it will drastically reduce, and we will start our normal life activities and educational activities very, very soon. Yeah. So hopefully you enjoyed a lot, and see you in the next time. I wish you all good day. প্লিজ কে জানি বলতেছো, প্লিজ বলো। কেউ বলতেছিলা আমি নেটওয়ার্কটা ক্লিয়ার না তোমার, প্লিজ আনমিউট করে কথা বলো। Maybe it's curtailed. It's cut down the network. So thank you all. Yeah. Okay. Allah Hafiz.